Hi, this is Dr. Chen, and this is a lecture presentation on Chapter 4, Sensation and Perception. So as you can see over here, the topics are going to be broken down into the visual system, the auditory system, gustatory system, which is your sense of taste, olfactory, your sense of smell, and touch. For today's lecture, in this pre-recorded lecture, we will be covering basically the essentials of sight. So one way to think about your visual system in terms of the processes involved in that, one useful way to think about it is that there is the um, sense part of the processing, and there is the perception part of the processing. The sense part of the processing concerns the hardware that we have to take the raw input from our environment, to process it in such a way and package it so that we can actually make some type of sense perception out of it, which is covered in the second part of the visual system here. Again, for this pre-recorded lecture, we will cover just the essentials of sight, which is the hardware that we have to process the raw information coming in from our environment in regards to our visual system. And we will touch base again this Thursday on perceptual processes and move on to the rest of the other types of your sensory systems. So first of all, when we talk about your visual system, we need some raw input and we need some light, basically, to see. But what is light? Well, light is a form of electromagnetic radiation that travels as a wave, as shown over here. And your light waves vary in terms of amplitude, so which is the height of the wave, as well as your wavelength, or the distance between these two peaks. Now, your amplitude determines your sense of brightness. The larger the amplitude, the brighter that light will seem like. Your wavelength is going to determine your perception of color. So now we got two very basic characteristics of light determined by the height of the wavelength, as well as the distance between two peaks in regards to that wavelength, which covers brightness and color. Now, in reality, light can also vary in terms of purity, which basically refers to how many different types of wavelengths are there present, or how varied the mix is in regards to the number of wavelengths. So you might have several wavelengths that correspond to different colors, such as blue and red and yellow, and they're all being registered in your retina. We'll talk about what a retina is in a couple of slides from now. But basically, in regards to purity, the more mixed the wavelengths are, the less pure the light is going to be. Another way to measure purity is saturation, which you can think of it as the relative amount of whiteness in a color. The more mixed your wavelengths are, the more white your color is going to look like. That means that the less mixed your wavelengths are, the less white your color is going to look like. Now, the less white your color is going to look like, the more saturated, as you can see, as it goes from left to right. So the less mixed your wavelengths are, or the less white your color is, the more saturated your colors are going to be. In other words, you will see more intense colors coming from a particular wavelength. So that covers the third property of light, which is essentially the different combinations of wavelengths that may be coming at you. And that determines the purity of your light. Now, when light passes through a prism, it is separated into component wavelengths, or the specific wavelengths that make up that light. What most people call light includes only the visible light spectrum, that is the wavelengths that only humans can see. As shown over here, the visible spectrum that we can see is captured in this continuum over here, and it's quite obvious that this only represents a slim portion of the total range of wavelengths that exists in reality. Now, interestingly, insects can see shorter wavelengths than humans can see in the ultraviolet range. Fish and reptiles can see longer wavelengths that humans can see in the infrared spectrum. So now that we briefly discussed the nature of wavelengths, we need to talk about how this input is converted into neural impulses that are then sent to the brain. So let's take a closer look at how this transformation is accomplished. Let's look at your main hardware that takes in this raw input, your eye, your eyeball. Well, your eyeball has two main purposes. Number one is to provide a house for the neural tissue that receives the light, which is your retina, which is towards the back over here. And the second purpose of your eye, not just to provide a house for your retina, but to also channel light towards your retina. So light enters through the transparent window at the front, which is also known as a cornea. And the cornea and the crystalline lens located behind the cornea form an upside-down image of objects that is being seen in reality. Now, you don't have to be too concerned that the image that actually sits on the retina is upside down. It doesn't really matter as long as your brain knows the rules for relating the positions on the retina to corresponding positions in the real world. Now your lens over here is made out of relatively soft tissue, and it can adjust through a process called accommodation. The closer the image or the closer the object to you, the fatter the lens is going to be to try to give you a clear image. Now this is the front view of your eyeball. Your iris, which is the colored portion here. This is the colored ring of muscle around the pupil, which is the black center of the eye. And your iris basically helps to constrict or dilate the size of a pupil, depending on the amount of light that is present in the environment. And this basically gives different amounts of light in. In a dark room, your pupil is going to dilate to let more light in, which sounds like a great idea. The downside to that is that the larger your pupils dilate, the less sharp your image is going to be. So it really is a trade-off, and you kind of need to find that sweet spot between letting in the right amount of light, but also not significantly compromising the vividness of the image that you see. Let's talk about some common conditions that we see in regards to the eye. So let's talk about nearsightedness first. So in nearsightedness, close objects are seen clearly, but distant objects are very blurry, just as shown in this picture over here. This is because the focus of the light from these distant objects falls a little short in front of the retina. So nearsightedness can occur in one of two ways. It can occur when your corneal lens bends light too much, or when your eyeball is basically too long, which makes the focus point fall in front of the retina. In farsightedness, where distant objects are seen clearly and close objects are blurry, the focus of the light is behind the retina. And this focusing problems can occur due to your eyeballs being not able to accommodate that well, or it could be because your eyeball may be too short for whatever reason. Either way, whenever the focus point falls behind the retina, you get farsightedness. Now, the retina is the neural tissue that lines the back surface of your eye. The retina basically absorbs light it processes images and sends visual information to the brain. Back when I was an undergrad, I was told by my biopsychology professor that there's a great deal of processing that happens in your retina. So your retina doesn't just absorb light, it processes a whole bunch of information concurrently, very quickly. So it's almost like a really very advanced computer chip in each side, in each of your eyeballs, that processes that information and condenses it in such a way whereby you can actually send bits of information that is usable to your brain. This box here may be a little bit misleading. This represents your optic disc, which we'll talk about in the next slide. But your retina basically lines the whole entire back part of your eyeball. All right. Now, axons from the retina to the brain converges on this part over here that is called the optic disc. So let me go back to the previous slide because that animation went by really quickly. So imagine your retina lining up the back surface over here. And basically at some point it needs to exit the eyeball. And this is the junction where the axons, which are the parts of a retina that is going to send information to your brain, this is where it exits your eyeball. And we're going to zoom in that section over there. There you go. Hopefully that made things a little clearer. So axons from the retina to the brain converges at what we call the optic disc, which is also known as a blind spot. It's basically kind of like a hole in the retina where the optic nerve leaves the eye. Now, the interesting thing about this is this. If an image happens to fall on this hole, this optic disc right here, you literally cannot see anything. It is what we call your blind spot. Now, most people are not aware that they do have a blind spot in each eye because each eye, and you have two, or at least most people have two, except if you're a pirate, each eye basically compensates for the blind spot in the other eye. And your brain somehow in the perceptual part of processing fills in the missing part of the image. So for the most part, we don't even realize that we have a blind spot, but we do. If you don't believe me, let's do this quick exercise with me. So to figure out your blind spot, I want you to close your right eye as you're staring at the screen and try to position your nose being in front of the X over here. And with your right eye being closed, focus your left eye on the X that is on the screen. Now move closer to the screen with your right eye closed and your left eye on the X. As you get closer, you will notice that at some point, the O is going to disappear. It's really interesting because you can move back and forth and repeat this as many times as you want. This is not a trick. It's an actual blind spot that we're dealing with. The point where the O disappears is when the light is shining directly on where your optic disc exits your left eye. And again, the reason why you can't see anything over there is because there are no rods and cones at the spot or the cells that are required to register light. So you cannot possibly register anything there. Now you may wonder, what do I mean by rods and cones? Well, that's going to be explained in this next slide here. Your retina contains millions of receptor cells that are sensitive to light. And these receptors, so think of this as this is the back of your eyeball. These receptors are actually located in the innermost part of your retina. So light actually needs to pass through several layers of cells, such as your ganglion cells, your amacrine cells, your bipolar cells. Light needs to pass through these layers of cells before it gets to the receptors that actually detect it. There are two types of receptors, rods and cones. Now rods outnumber So rods are the longer ones compared to cones. They're the most stubby ones. Rods outnumber your cones by a huge margin. We're talking about 100 to 125 million rods to only about maybe 5 million cones. Now, rods play a key role in night vision because they're 100 times more sensitive than cones in dim light conditions. In addition to that, rods also play an important role in peripheral vision because your rods greatly outnumber your cones, especially in the periphery of your retina. On the other hand, cones play a key role in daylight vision and color vision. Cones don't do really well in dim light, but in bright light, they provide the sharpness and details that rods cannot provide. This explains why things appear black and white in the dark, because your cones can't really work very well in dim light conditions. Now, your cones are also most heavily concentrated in the center of your retina, and it quickly falls off in density towards the sides, where your rods basically try to make up for that. And your fovea is a tiny spot in the center of your retina, that sweet spot that contains only cones. And it turns out that visual acuity is greatest at this spot. If you've ever seen someone maybe try to tilt their head downwards and try to look up when they try to focus on something, what they're trying to do is they're trying to adjust the light so that that image is falling dead center on your fovea, which is where visual acuity is greatest. So that's kind of what people are trying to do when they tilt their heads in various ways to try to get that sweet spot where you can get the most amount of vividness and detail. So remember, in terms of receptors in your retina, you got rods and cones. Rods play a key role in night and peripheral vision, and cones play a key role in color and daylight vision. Now, you've probably noticed that when you enter a dark theater on a bright day, you'll probably stumble about for a little bit, right? But within minutes, you can make your way quite well in dim light. That process of adjusting to a darker surrounding or even a lighter surrounding, it's called adaptation. And if you're talking about adapting to a darker surrounding, that is called dark adaptation. As this figure here shows, as time passes from 0 to about 30 minutes, you require less and less light to see, because this y-axis represents the threshold for light detection. Now, cones, remember that cones play a key role in color and daylight vision. Interestingly, cones adapt more rapidly to darkness compared to their rods, but rods adapt better in the long run. So in the beginning, your cones really do help you adapt to the dark really quickly, but that's only so far that you can go. After which, you need to depend on your rods, and it takes about 30 minutes for you to basically complete dark adaptation. A really funny story that one of my biopsych professors had told me was that he said, if you've ever wondered why pirates have a patch on their left eye or right eye, it's not so much because that someone poked them in the eye. It's because when you're moving, say you're trying to loot another ship or something, or maybe your ship is under attack by someone else, and if you need to transition really quickly between the top of your ship to the bottom or the hull of your ship, that requires you to adapt to light and dark really quickly. Well, how do you do that? Well, by preserving one of your eyes, so that one of your eyes is fully adapted to dark and the other eye being fully adapted to light. You can switch both eyes as needed, so you can go up and down and transition from light to dark really quickly. If you don't believe me, you can try this on your own. The next time you get up in the middle of the night and you need to run to the restroom and you don't want to be stumbling about in the dark right after you've done your business, close one of your eyes. And yeah, so make sure you close one of your eyes before you switch on the lights in the bathroom. And once you're done, switch off the lights and then open that eye again that was closed. You'll find that you can almost instantly adapt to the darkness that is in the rest of your home. So try that. It'll be something fun for you to do if you've never done that before. As mentioned before, there's a great deal of information processing that happens in your retina. Information from as many as 130 million rods and cones converge to travel along only about 1 million axons in the optic nerve. That's like the ultimate zip file application, because you're trying to condense 130 bits of information into 1 bit of information. So this means that along the way, when light first registers in your rods and cones and it gets sent out through your bipolar cells and endocrine cells and ganglion cells, these cells over here, these layers of cells here, integrate and compress signals from your receptors so that all of the information that's coming from the rest of your retina can be compressed and pushed through your this small little space that you have here at the back of your eye, which is your optic disc. The receptive field of a visual cell in your retina, that is when stimulated, affects the firing of that cell. We'll come back again to the concept of receptive fields when we talk about your touch sensory system later on. Although the retina does do a lot of information processing as a sensory organ, we still need to process that visual input in the brain. And so this diagram over here shows you the visual pathways in your brain. Now this can seem a little bit tricky if you don't pay attention to what's going on over here. So see if you can follow me through this, and I have a follow-up slide to make sure you actually get this. This represents your visual field. This is your left eyeball and the right eyeball. Let's take information on your left visual field, right over here. So information that is in your left visual field is going to be registered on the right retinas on both your left and right eye. And this is where I think some people get confused, because they think that all light on your left visual field only gets registered in your right eye. That is incorrect. That's not how light travels. Remember, light travels in a straight line. Light doesn't care which eyeball it goes into. So any light that is in your left visual field is going to be registered in the right retinas on both eyeballs, and that information is going to go down this red pathway over here and then gets further processed in the right hemisphere of your cortex. So in a nutshell, everything in your left visual field gets registered in the right retinas on both your eyeballs, left and right, which then gets transferred to your right hemisphere for further processing. It is the inverse for everything that is on your right visual field. Everything in your right visual field is registered on your left retinas on both eyes, which then gets sent down the line and processed in your left hemisphere in your brain. Now, the part over here where we transition from your left side of your brain to the right side of your brain is what we call your optic chiasm, where fibers from the inside half of each retina crosses over to the opposite side of each brain. Here's another way to look at this in case if the previous diagram is not making it clear again. Anything that is on the left visual field is registered on the right side of the retinas on both eyes, which then gets sent down the right hemisphere for further processing. Everything that is on the right side of the visual field gets registered on the left retinas on both eyes, which then gets processed down the line in the left hemisphere. So let's look at this diagram again. After reaching the optic chiasm, the major visual pathway projects through the lateral geniculate nucleus, which is this and that. So I really should have another line over here. The major visual pathway projects through the lateral geniculate nucleus in the thalamus and onto the primary visual cortex. And so shown by the solid lines, the red line and the green line right here. The second pathway detours through the superior colliculus and then projects through different areas of the thalamus and basically ends up in the primary visual cortex too, as shown in the dotted lines. So we got two major pathways here after information passes through your optic chiasm. One through your lateral geniculate nucleus, the other through your superior colliculus. The second pathway, the one through the superior colliculus, appears to be involved in the coordination of visual input with other sensory input. So if you're into sports, for example, and you're running down the field and you're trying to catch a football, you are trying to coordinate your visual input and other sensory inputs that you have, such as your sense of touch, your sense of hearing, and other basically senses to coordinate your movements to be able to catch that football and score that touchdown. Really interesting stuff. Well, at least I find it interesting. I hope you find it interesting too. Now, all visual input, you might have noticed in the previous slide, reaches the occipital lobe of the cortex at the end of the day. So the occipital lobe of the cortex is basically the back part of your brain right here. And researchers have investigated how the cortical cells in your visual cortex right here, which is located in your occipital lobes, how your cortical cells right here respond to light. And how they do that is by placing microelectrodes in the visual cortex of animals, such as cats, to record the action potentials or the activity of individual cells. We'll learn more about how neurons communicate with one another in Chapter 3. We'll basically do that after we're done with Exam 1. But for now, suffice for you to understand, I think, that the way in which neurons in your brain communicate with one another is by firing electrical impulses, which is shown here by the vertical lines here. So the more vertical lines you got, the more active that cell is going to be, essentially. So what researchers initially did was they would flash spots of light in the retinal receptive fields right here. But the weird thing is they weren't getting any activity in the cells in the visual cortex, as shown here on the oscilloscope. And that perplexed researchers for some time because they're like, why aren't the cells here responding to light? It was actually David Hubble and Thorsten Wiesel that discovered the reason for this lack of response to these shades of light. And they actually discovered it by accident. What they found was that the cells in the visual cortex would not respond to these spots of light over here, but they would respond to specific shapes such as lines or edges, and down the line in the visual pathway, somewhat even more complicated stimuli rather than small spots of light. And they were actually awarded the Nobel Prize for their groundbreaking work in this area because it had perplexed lots and lots of people for some time. So this is an example of how one particular neuron is firing action potentials, which is a measure of its activity when it sees different shapes. So as this cat is shown various stimuli, a microelectrode connected to the visual cortex here records the firing of the neuron. So remember, with more lines represent more rapid firing, more activity. So we can see here that a vertical line elicits a lot of activity, a lot of firing. A horizontal line, on the other hand, does not elicit a whole lot of firing. A line tilted diagonally elicits some level of firing, more than when the line was horizontal, but less than when the line was vertical. So after a visual input is processed in the primary visual cortex, signals travel through two streams that has sometimes been characterized as the what pathway and the where pathway, the ventral stream versus the dorsal stream. The ventral stream processes details such as what objects are out there. So it basically processes form as well as color. So if there are any cells in here in the ventral stream that is damaged, you're gonna have some problems trying to identify form and color. On the other hand, the cells in the dorsal stream process details pertaining to motion and depth. That's why it's called the where pathway right here. And what they found was that, remember this little study that they did with cats and they put it in the primary visual cortex? Well, they basically placed the microelectrodes in various locations in the ventral and dorsal stream. And they found that as signals move further along the visual processing system, neurons became even more specialized and fussy about what turns them on. And the stimuli that activates them becomes more and more complex as you go down the pathways. In monkeys and humans, for example, there are neurons further down the line that respond to pictures that basically resemble a face. If you think about how a face looks like and the basic shapes that make it up, it's basically much more complicated than a single line. And so it's interesting that there are specific neurons that would only fire when it sees a combination of shapes that look like a face and nothing else. And that may have some implications on the evolutionary importance or the evolutionary adaptiveness of being able to have that ability. So remember the ventral stream and the dorsal stream and what they basically code for in your visual system. So being able to detect objects in motion is all great, but can you imagine a world without being able to see in color? Many animals have little or no color vision, and it wasn't that long ago that, photos in movies and TVs were black and white. However, it has been suggested by some theorists that color vision evolved in humans because it improved their ability to find food, to spot prey, and to readily recognize predators. So let's talk about the different ways in which we can mix colors. As this color solid demonstrates, people can perceive many different colors. Most of these variations are the result of mixing a few basic colors, and there are two kinds of color mixing. The slide over here talks about additive color mixing. The next slide talks about subtractive color mixing. Additive color mixing works by superimposing lights, putting more light in the mixture than exists in any light by itself. So if you shine red and green and blue spotlights, you'll find that basically that gives you a whole lot of mixture that gives you a white color. If you mix a red and green wavelength and superimpose those two wavelengths, you get the yellow and all the different combinations. Subtractive color mixing, on the other hand, works by removing wavelengths of light, leaving less light than was originally presented. So for example, if you were to mix red and blue and green pigments, like paint for example, you'll find that you get black. Paints yield subtractive mixing because the pigments absorb most wavelengths and then selectively reflect back specific wavelengths that give rise to particular colors. So if you look through a sandwich of, say, red and blue cellophane filters, they will block out certain wavelengths, and the middle wavelength that will be reflected back will look purple. Now, the human process of color perception in a visual system parallels additive color mixing much more closely compared to subtractive color mixing. Let's talk about two major theories of how we see the different colors of the rainbow. In the middle of the 19th century, parallels between additive color mixing and human color perception inspired what we now call the Trichromatic Theory of color vision. This theory holds that the human eye has three types of receptors sensitive to the specific wavelengths associated with red, green, and blue. So according to this model, people can see all the colors of the rainbow because the eye does its own color mixing by varying the ratio of neural activity amongst these three receptor types. So if we see, for example, if you have cones, for example, in your retina, that are able to activate when a wavelength that corresponds to green comes in, that cone is going to fire more action potentials, thereby giving the signal that you're seeing green. If you want to see yellow, then we need a mixture of wavelengths such as red and green, so that you got now two cones firing at about the same rate, giving you the impression that you're seeing yellow. So again, according to this model, people see all the colors of the rainbow because your eye, through its cones, does its own color mixing by varying the ratio of neural activity amongst the three types of receptors that roughly corresponds to these basic colors. And in fact, most of what we know about color blindness matches fairly well, not everything, but it matches fairly well with trichromatic theory. So we know, for example, that the vast majority of colorblind people are what we will call dichromats. That is, people who make do with only two color receptors. And there are three types of dichromats, and each type is insensitive to one of the primary colors that is talked about in the trichromatic theory of colors: red, green, or blue. So most people are either insensitive to red, insensitive to green, or insensitive to blue, although the insensitivity to blue is more rare compared to insensitivity to red and green. So the three common deficiencies that we see supports the notion that there are three sets of receptors for color vision. So although trichromatic theory explains some facets of color vision well, it did have some problems. Take for example complementary after images. So trichromatic theory cannot explain complementary after images, and so what are complementary after images? Well, there are two ways that you can think about it. First of all, complementary after images are pairs of colors that produce gray tones when mixed together. So if you mix purple or violet blue and yellow, you get gray. You mix red and green blue, you get gray. And so on and so forth, as shown in this diagram over here. Another way to define after images are basically the color that you see when you stare at something long enough and then you look elsewhere. So you typically see like an after image or a visual image that persists after looking at something for a really long time, and typically that after image has a different type of color than what you were seeing. So if you're staring at a red apple for a really long time, for example, and it doesn't really have to be really long, 15 seconds will do, and you look elsewhere, especially if you look at a blank white surface, you'll find um, a green-blue after image that comes after that. So if you don't believe me, we can try to do this exercise together. So zoom in right here. So what I want you to do is stare at the white square in the middle of this diagram over here and try to stare at it for about 15 seconds. You want to try to not refocus your eyes if at all possible, and just stare at it for 15 seconds, the white the white square. So I'll give you 15 seconds to do that right now. Did you see the after images? So I hope you were able to see that, because I definitely see it looking at it for a really long time. The red square probably would have produced a green-blue type of after image. The yellow square would have produced a more violet blue type of after image, and so on and so forth. So a trichromatic theory, if truly the way in which we see color is mostly because of those three cones, that theory itself would have quite a bit of difficulty trying to explain why we see these after images. This is why researchers started looking into this theory called the opponent process theory. So what is the opponent process theory? The opponent process theory postulates that color perception depends on receptors that make antagonistic responses to three pairs of colors: red versus green, yellow versus blue, and black versus white. So for example, staring at the red image for 15 seconds causes your red opponent cells to become really tired, right? And when you shift your focus to a blank surface, those cells that were firing in response to the red wavelength are no longer able to fire, not as much at least, so only the opposing green cells will continue to fire in response to that visual stimuli. And as a result, you will see a brief after image of green if you're staring at something that's red, and it'll be vice versa if you did it the other way. If you stare at something black for a really long time and look away, you'll see the white after image. If you stare at something blue, you'll see the yellow after image, and so on and so forth. So how do we put all this together? Well, research has identified three types of cones. And actually, before I go into more detail about this, basically there is some truth to both these theories of vision or color vision. Let's talk about what we found out so far that supports trichromatic theory. So research has identified three types of cones in your retina that has varying sensitivities to different wavelengths of light. As this graph here shows, and the x-axis represents the wavelength, and the y-axis represents the receptor sensitivity. Excuse me. You have three types of cones that correspond roughly to red, green, and blue, as predicted by trichromatic theory. In reality, it's probably more accurate to refer to them as cones that are sensitive to short, medium, and long wavelengths. But in terms of talking about these various things, most people understand quicker when you talked about red, green, and blue. Um, in addition, so we've also found some evidence supporting the opponent process theory. We found that cells in your retina and further down the line, especially so in your thalamus and in your visual cortex, there are cells that respond in antagonistic ways to red versus green and to blue versus yellow. So, for example, there are some ganglion cells. If you remember back to that picture when we zoomed in your retina, there are some ganglion cells that are excited by green and inhibited by red. And then there are other ganglion cells that work in the opposite way too. So in summary, what most researchers now believe is that the receptors that do the first stage of processing your cones seem to follow the principles outlined in trichromatic theory. In later stages of processing, in the retina and down the line in the pathways in your brain, they seem to follow their principles of opponent process theory. And that concludes this really brief lecture on the essentials of sight. Um, when we meet again this Thursday, we will cover the rest of perceptual processes. Basically, how do we take all this raw information and actually make sense of it as a whole? Then we'll talk about your auditory system, gustatory system, olfactory, and your touch sensory system. So we'll obviously not go into the same level of detail when talking about your other sensory systems, but um, we'll at least cover some of the basic facts related to these things. I hope this lecture made sense. Um, I hope that you'll have a good time answering the online forum as well. Please let me know if there is anything that you don't understand, and I'll be more than happy to talk to you about anything pertaining to this chapter. Thank you, and have a good rest of the day.